guys, this is Mr. Brenton here, and we're going to be talking about osmosis. What is it? When does it occur? And how do we predict osmosis? So, what is osmosis? Osmosis, let me go here, is the diffusion of water through a membrane. Water through a membrane. Well, when does that occur? If you remember our discussion about diffusion, diffusion occurs from areas of high concentration to areas of low concentration. Well, that does play a factor here, and that is a rule here for this too. And we're looking at concentrations of water, but we also compare them to the concentrations of salt. And sometimes it's easier to look at the salt as opposed to the water when we're predicting what will happen. And the rule basically is that water diffuses towards the salt. Okay, and the reason why is salt will actually grab a hold of water and basically lock up water, so it's not free moving water anymore. And so the water that's moving freely from that area of high concentration of highly concentrated water to areas of lower concentration of water where more of that water is locked up with the salt. This also explains why salt helps ice to melt. It locks up the free amount of water, so it lowers the concentration of water that way, and where the salt is. And then so the water is more likely to melt than it is to freeze back into water. Okay, so we'll get to that part a little bit later. But let's get to some examples. So let me just scroll down just a little bit, and let's go over one example that we're talking about here. Let's draw a cell with a cell membrane, and in this example we're going to say the cell has 95% water and 5% salt. That's about normal. Okay, and what we're going to do is we're going to put it in a salty environment. Let's say we put an environment with 10% salt, so 90% water and 10% salt. Okay, so if you remember our rule is that water will diffuse towards the salt. Well, in this case it diffuses towards the greater amount, a greater concentration of salt, or a greater percentage of salt. So in this case, water is going to diffuse from inside the cell to outside the cell. So let's draw a few arrows here. So this is the direction that water is taking. Okay, so more water. So if you can imagine, this cell is going to be losing water. So water diffuses out of the cell. Out of cell. Okay. So in that case, let's just get my pen back here. Out of cell. All right. So now, or we can say the cell loses water in this situation. So basically, if you put a cell into a very salty, like a brine type situation, it's gonna dry it out. It's gonna lose water, and we call this solution a hypertonic solution. Hypertonic. Okay, solution. That is where you have a salt solution, like salt water. Okay, so in saltwater solutions, we say the cell loses water. Okay, water will diffuse out of the cell because remember, that salt locks up that free water, so it reduces this percentage of water, and so you have a lower percentage of water outside the cell, and you have a higher percentage of water inside the cell. So there's more free moving water, and there's more of a chance of the water leaving the cell than entering the cell. And it doesn't mean that water doesn't enter the cell. It can, but again, there's more water exiting the cell than entering the cell. So that overall the cell will lose water. Okay, let's move on to the next example. Let's see. Hang on one second. Move this down. Oops. Okay, so example number 2 is going to be, let's draw our cell again. Still 95% water. Oops, 95% water. And 5% salt. So we're sticking with that. But we're going to change this solution now to 100% water. So no salt. So 100% water, 0% salt. Well, again, we said water goes toward the salt. So in this case, we're going to have more water entering the cell. So we're going to have water. Again, that's where we're going to have more of our water entering the cell. So it is possible that we can have water, and we do, since this water molecules are moving randomly, there will be some water that does leave the cell. But again, more water is leaving the cell than, I'm sorry, more water is entering the cell than leaving the cell. Okay, so, all right, so let's put that down. So water, we'll say the cell gains water. Oops, yeah, we can put that there. Oops. Okay. All right, and this can be a very dangerous situation for some cells, like for instance, if our red blood cells are placed in distilled water or pure water, they would actually gain enough water and the cell membrane would rupture and would destroy the cell. All right, so let's see, what type of solution is this? This is a hypotonic solution. So hypo meaning less, tonic meaning salt. So basically this is something like distilled water would be a good example. Distilled water is an example of a hypotonic solution because that's 100% water. All right, let's take a look at the last example. Oops. You can imagine what that one's going to look like. Example number 3. Again, let's draw our cell, and remember we're going to stay with the same percentage of 95% water and 5% salt. Well, if the first example we had a greater amount of salt on the outside, then on the second example we had less amount of salt, well, you think of the only other example we could possibly have is, you guessed it, they're equal. So in this solution we would have, surrounding the cell, all this water, it would be 95% water and 5% salt. Well, what's going to happen there? Neither side of the membrane has a greater amount of salt. Well, you guessed it, it would be equal, And so for every molecule you have entering the cell, you would have another molecule probably leaving the cell. So the cell does not gain water and it does not lose water overall, but water is entering and leaving the cell equally. So it reaches that dynamic equilibrium. That's where this is. So dynamic equilibrium is that the cell does not gain or lose a net amount of water. Okay, so but water does enter and leave the cell in equal amounts because water is always moving, but it reaches that dynamic equilibrium because the concentrations of salt are equal. Okay, so concentrations of salt are equal. Put that over here. This is what's called an isotonic solution. Is equal salt on both sides of the membrane. Okay, so basically we see the inside environment of the cell has 5% salt. The outside of the environment has the outside environment of the cell has 5% salt as well. So since the salts are equal, the cell is not going to gain or lose water overall, even though water is entering and leaving the cell just because of the random movement of molecules and them passing through the membrane if they're going in the right direction. This is how saltwater fish survive, and there is also some saltwater invertebrates that, why don't they gain water? Or I should say, why don't they lose water? They're in a salty environment, and their salt has to, inside their cell, has to equal the salt in the environment outside the cell. And that's an important situation there. And you can even see these extremophiles where these bacteria live in very, very salty conditions, and they are able to survive by matching the salt inside the cell and outside the cell. And that's going to be our last example and of why water diffuses through the membrane, and this is called osmosis, and we have to understand that to understand how we can gain water or lose water. Okay, so this is it, and I hope you learned something, and we'll see you in class. All right.